I want to say thank you, everybody, for doing the presentations today. Appreciate it. Today is my pleasure to welcome Dr. Anmar Khadra. He is currently a full professor at the Department of Physiology and a co-director of the Center of Applied Mathematics in Bioscience and Medicine at McGill University. His research focuses on developing mathematical and computational models to understand complex biological systems via the application of nonlinear dynamics, stochastic processes, and computational methods to study problems in neuroscience, in immunology, and cell biology. Today, he's going to give us a talk about the intrinsic dynamics under synaptic bombardment. So go ahead.
Thanks very much, Nate, for the invitation. Can I close the door? Is that okay? Because I found it a bit distracted. Thanks, Yangyang, for inviting me to give a talk here. So today I will be telling you a little bit about a new direction. Actually, it's not new anymore, but at least a direction that I've been exploring for the last couple of years, translating intrinsic dynamics of neurons to their circuit behavior, how to scale up single-cell electrophysiological properties to their circuit dynamics, and of course link it also to the system-level behavior, how these dynamics could affect the behavior of the animal while they're performing a task. Okay, by the way, this is downtown Montreal from the mountain, and this is McGill campus here, and my office is right there. This is my office, overlooking the mountain. And the RCMNS will be held in that building in a couple of months from now. Okay. All right. So as you may know, typically we record the electrical properties of neurons at the single cell level using different methods like whole cell configuration, for example, or patch plan. And that will tell us a little bit about the intrinsic properties of these neurons. And at the same time, experimentalists, they also use different kind of methods to record from these neurons in vivo while the animal is performing a task. And essentially, the type of properties that you record from here will be influencing the results that you see here, but typically the properties that you see here are not always consistent with what you see in vivo. And the question is, how do we bridge between the two? How do we actually understand how they kind of, how can you scale up this to this? And that's exactly what I'm trying to basically cover in this talk. And that's essentially what I just said right now. And now for the intrinsic properties, we know that these are dictated by the different ionic currents that are expressed on these neurons. While in vivo, they are affected by the extrinsic properties, how they are interacting with other neurons within the network, and that actually affects their behavior within the circuit. And what adds another complexity to this whole thing is that there is also heterogeneity. You've been working on this for quite some time, and I'm bringing it up here right now, is that, not just that different types of neurons have different properties, but even within the same class of neurons, you have quite a bit of variability in terms of behavior, how these neurons behave. And the question is, how do we actually link or explain all of these things together within one class? We want to basically link in vitro to in vivo and also account for the heterogeneity that exists within the same class of neurons. Now, let me give you an example, an animal model that we use in our project. And that animal model is the vitro electric fish. And we focus on one particular type of neurons. These are the pyramidal cells in the electrosensory lateral line lobe, or VLL. These neurons typically receive stimulus from neurons that are on the skin. These are the electric receptor afferents located on the skin, and they release an electric field, like an electric field called VOG. And at the same time, the ELL transmits signals to higher brain regions, like the circularis and nuclear tegmentralis. And I would like to emphasize that there is also a feedback coming from the MP back to the pyramidal cells. And let's keep that in mind, because that becomes really important later on, the feedback coming from higher brain regions back to the pyramidal cells in the ELL. Okay, so we focus on these neurons. You could think of these neurons as being like the Purkinje cells in the cerebellum, which are responsible for motor control, So they share quite a bit of similarities to other cell types in higher primates. Okay. Now, if you record from these cells in vitro, you'll see that they exhibit bursting dynamics that looks essentially like this. This is in the dendrites, this is in the soma, where you generate clusters of action potentials together, and this depolarization after potential that is a signature of these recordings, these burst firing. And people have investigated this quite a bit in the literature, and they developed even model, and I alluded to this a bit later on. But at least when you compare the in vitro recording in these cells to those that are recorded in vivo, you'll see they're quite distinct. You don't have the gap that you see in vitro recording, and also the clusters of action potentials, these bursts, are much shorter. You could get 2 to 4 spikes per burst, but these ones in vitro could be much more spikes than that. So there are differences, and the question is, how do you reconcile the two? Again, I told you about the complexity that is added due to heterogeneity. Now, if you use neuropixel probes and you record from these neurons, while the animal is actually immobile but fully conscious, and you record from all of these neurons from ELF, you see there's quite a bit of heterogeneity in the firing patterns of these neurons. In fact, some of them can even switch between bursting and these kind of isolated sparking events within the same recording, and there's quite a bit of variability. So the complexity becomes quite significant when you combine all of these features together and try to understand what's happening. Okay, all right. So one thing I would like to mention, at least in the context of these intercellular recordings that are done in vivo, which I highlighted that there could be between 2 to 4 spikes per burst, is that if you look at the ISI distributions of these recordings, you generate this kind of bimodality. So let's say that you look at this spiking train that you hear from one single neuron, and you look at the ISI distributions generated by these neurons, you generate that bimodality. So within that bimodality, you could identify 2 subclasses of firing patterns based on the minima here that you see here. You generate the burst from those isolated spikes within the same recording. And you could use that threshold to basically distinguish the burst from the isolated spikes. And that's actually a strategy. That's the threshold I'll be using quite frequently when I do most of my analysis of these recordings. Okay. All right. So this is some important features that we have observed, at least in vivo, in these cells. But that brings up a very important question, and that question is, what role are these bursts playing? Now, there's another layer of complexity that is added on top of everything that I've mentioned already. I have highlighted, first of all, the in vivo versus in vitro. I highlighted heterogeneity, and I'm bringing up this extra question of what is the role of these bursts, at least in neurocoding, when it comes to these animals when they're performing, when they're awake and functioning. So that brings me to the outline of my talk. The first thing that I would be basically analyzing is how in vivo and in vitro dynamics could be linked together. We'll discuss the sources of heterogeneity and use some experimental conditions that we can use to validate some of the predictions that we're making here. And finally, I'll explore the role of bursting in neurocoding. All right, so let me give you an overview of some of the literature. Dorian et al. Published a paper in 2001. That model was a two-compartment healthcare hospital model that linked soma to the dendrites. And through that two-compartment model, they managed to actually generate what we call ghost bursting. Ghost bursting that is responsible for generating that type of bursting that you see in the recording that I highlighted with in the soma versus in the dendrites. Okay. But I just mentioned earlier that in vitro, in vivo, the bursting is quite distinct. You have 2 to 4 spikes per burst, and you don't have the gap that you see in vitro. One hypothesis that was suggested, at least in the literature, that distinguishes between these two is that the SK channel, small conductance potassium channel, could be involved in generating these differences between what you see in vitro and what you see in vivo. And then you might ask the question, are you saying that the expression level of this channel in this condition is different from here? The answer is no. It's not different between the two, but calcium could be different between these two conditions. These neurons receiving synaptic inputs while these neurons are not. And these synaptic inputs could drive changes in calcium, and that changes in calcium could affect the SK channel. And that's what we're arguing. And I'm going to basically use mathematical modeling to explain the switch from here to here through the SK and calcium dynamics. So how do we do this? Again, I'm going to adapt the Touboul model, but improve on it to account for this, the role of the SK and calcium. So again, it's a two-compartment model. One of them is the SK channel, that's a calcium-dependent channel, and the NMDA receptors, and the NMDA receptors reflecting a synaptic bombardment. They are activated by the synaptic inputs that these neurons are receiving. Okay. Now, the synaptic input is modeled by the NMDA kinetics is modeled using this Markov model, which is adapted from Justitial. And again, we have to account for calcium dynamics within the dendrites. And the calcium dynamics, you have calcium influx through the NMDA receptors, calcium-induced calcium release through the RP3 receptors in the ER. And then you have, of course, the leak and the pumps, the MCA pump and the Sreshta pump that cover the dynamics of calcium within the dendrites. Okay, so the calcium dynamics entry through the NMDA could activate the SK channel, and that is that activation that was missing in the Touboul model is what we argue is distinguishing between the in vivo versus in vitro. Now, what is the output of the model based on this mechanism? This is essentially the output of the model with synaptic bombardment. You end up with the voltage in the dendrites, the voltage in the soma, and that looks very similar to what you see in vivo. And this is calcium dynamics, which within the physiological range that you expect for any cell in your body, that it doesn't exceed more than 100 to 100 nanomolar to 1 micromolar. Okay. Now, visualization is one thing, and validating that this thing, the statistics of that signal that is generated here, matches what you see in vivo is another. And basically, we wanted to validate this, to make sure that the model captured the essence of everything that we know about these cells. To do this, we developed a pipeline. That pipeline essentially is a maximum likelihood algorithm that will allow us to match the model to the data. Now, we have, remember, in vitro data and we have in vivo data. Both of these together should be matched to the model simultaneously. So we need to constrain the model essentially. So when it comes to in vitro, we looked at different features within the action potential that is recorded in these cells within one single recording with multiple spikes. And we looked at these different features. One of them is the temporal, down course on the upstroke and downstroke, the peak amplitude, AHP, and the AVP. All of these features that are adapted from the Allen Institute, we decided to actually see within a spike train how they match between the data and the model. So we constrained it to make sure all of these features are actually equivalent between all of the data and the model. So we match these in vitro, and then we asked how we can actually match it with the in vivo recording as well. And that's essentially what we did. We used the same pipeline, and now we're using the ISI distribution from the data. ISI distribution from the data in terms of different features that we have to account for, like the mean burst fraction. Remember, we have the threshold for the bimodality, where that allowed us to identify the bursts, the mean ISI, this is the mean frequency, mean ISI, median ISI, standard deviation, and coefficient of variation. And you could see that there's a very close match between the data and the simulations. So we constrained the model in vitro and in vivo to make sure that it captures everything essentially in the data. Okay. Now that we know that the model has been constrained sufficiently enough to both of these classes of data, we can now use it to do the analysis. First of all, we did study the kinetic dynamics. So in the absence of noise, in the absence of the synaptic bombardment, and we tried to see what happens as you change the applied current to the cell. Now, 3 different regimes of behavior can be observed. This red line here corresponds to stable equilibria, and that's essentially represents the quiescence that could be observed in the cell. The envelopes here, this is the maximum and minimum of oscillations, and that emanate from a hub application that corresponds to tonic firing. But if you zoom in into this small gray area, this is where all the exciting dynamics happen. Right there, you have period doubling modification, where you enter the bursting regime. And that bursting regime here, you end up with doublets, you end up with ghost bursting with multiple spikes, and every burst with number of spikes generate a branch. So these branches here could actually overlap. And you have infinite number of branching points that correspond to bursts with different number of spikes within this regime. Now let's go back to the question I asked before. We talked about escape channel and calcium dynamics. Escape channel and calcium dynamics that distinguish between these two features, between in vitro and versus in vivo. Can we study the dynamics relative to these conductances? So we did this, and we changed not just I applied, but the conductance of the SK and the conductance of NMDA that fluxes calcium into the cell, and we tried to analyze what happens to all of these parameter regimes that you see there. The quiescence, the tonic firing, and chaotic bursting or ghost bursting that you see there. And you see that you can actually see the different regimes and the type of behaviors that you can observe, just like we expected to see in the mechanistic dynamics. So in this regime here, for example, you can just generate doublets. That's number 3. You can here, this is by the way, this is a multi-stable regime here where you could have multiple activities. So in 3 here, you have doublets, but you could also have somewhere here this chaotic bursting that you see there. And with the NMDA, you still have something like this. Okay. So essentially, you could explore different regimes of behavior that could mimic the behavior that you see in vitro. Okay. And if you basically try to plot the firing rate as you change the I applied, you'll see the kind of chaotic dynamics that you expect from this model. You could basically go through these bifurcations, and which essentially consistent with what you see at least in the analysis that we have here. All right. Now we have a model. And we thought at the time is that this model, obviously I'm going to show you later on that we can use it for further analysis. But before we moved on to do that analysis, we asked the fundamental question, is it possible to actually simplify this model and generate a phenomenological model that can capture all the dynamics that we see here, that one can use actually to develop neural networks? And of course, one thing that and I would like to emphasize is that if you look at the ghost bursting here, there's one feature that you can notice that the interspike interval within the burst gets shorter and shorter, which is atypical of any bursting dynamics that you see in the literature. Ghost bursting has really this unique feature is that the frequency increases rather than decreases within the burst. In every other bursting patterns that we know in the literature, the opposite would be true. So we wanted to solve that issue. To do this, we came up with a simplified model that is based on the Anmar model. Remember, the Anmar is a 3-dimensional model consisting of a voltage variable, the recovery variable, and a slow adaptation variable. These 3 variables would generate bursting that are typically involves a frequency that decreases within the burst rather than increases within the burst. So the opposite of what we want. So to resolve that issue, we added another adaptation variable called U here, which essentially does this effect and changes the dynamics to make the frequency of the firing to increase within the the rate of the firing increase within the burst rather than decrease. And we add another extra feature, which is sorry, I'm not showing it here. Here we basically try to fit the model to the data. But I'll mention that later, is that the HMR model itself switches dynamics from quiescence to burst state. But the ghost burst state goes gradually from quiescence to tonic firing to burst state. So after we fitted the model here, we tried to see if it can capture that dynamics as well. And this is what you see here. So essentially very similar to what you see as well in the ghost burst model. So we have again the quiescent, the tonic firing, and within this regime is the interesting dynamics where we have the periodic bursting regime, similar to what you see in the data. What you see also in the ghost burst model. You have the doublets and you have also the ghost bursting behavior that you see there. And as you can see here, that with the synaptic bombardment, you get an excellent match between the data and the model itself. Whether depending on the feature that you select from the ISI. Okay. And again, similar, when you look at the firing rate as a function of I apply, you get also patterns similar to what you see in the ghost burst model. So yeah, I mean, the features are captured, and the question becomes, what is actually producing that heterogeneity? Remember, we talked about the in vitro, in vivo, and that extra layer of complexity, which is heterogeneity involved in the data. Can the model, the simplified model, or we call it the modified HR model, can it actually explain what is the sources of heterogeneity that is coming from the data? To do this, we quantified what we call Touboul indices, which is essentially a metric that would help us determine the sensitivity of the model and the parameters. How much do you need to change a parameter to capture the variability you see in the data? Okay, so we looked at all the parameters simultaneously and we realized that there are two key parameters here that are contributing most to the heterogeneity that you see or the variability that you see in the data. And these two parameters are the synaptic bombardment, this is the noise intensity, as well as this adaptation parameter, a slow adaptation parameter within the dynamics. Okay, so two of these are key in generating the heterogeneity that we see within the data. Okay, so that kind of insightful, and we want to use this eventually. But before I move on to using that information, I just would like to emphasize that without noise, coming back to this idea, like, this is important to capture the variability. So if you remove the synaptic bombardment, you essentially bring the model back to being deterministic. If you do this and you compare the deterministic distribution of ISI versus the data, they don't match at all. So you need that synaptic bombardment to make them match. Emphasizing the importance of this parameter in generating this match between the two. So this is key in generating the variability within the data. Okay, yeah, go ahead.
What is the synaptic bombardment? What does it mean in the model? Is it increasing calcium to NMDA or?
And this is here in this scenario, in the physiological model, it's just a 1/f noise that is added to the voltage gradient. 1/f noise. Okay, but in the physical model, it's essentially we included the NMDA activation of glutamate release, and I'll show you the specific parameters that present these. Okay, so it's more biophysical than what you see here. Does that answer your question?
If this is noise for a particular variable, you have noise in Z, right?
No, just one variable.
They have sigma Z.
Oh, I see what you mean. Okay, yeah, other variables that are added, exactly. So this is the adaptation variable.
Yeah.
The adaptation variable, coming back to this, exactly. Okay, sorry. All right, now that we made a prediction about these two parameters, now let's go to the biophysical model. Remember, they may basically match the interspike interval data or the distributions that you have from the data, and we had an excellent match. Just to refresh your memory, the R value was really high, and these are two sample cells to show you how the distributions match exactly, and these are other metrics that show really like a great match between the two. Yeah.
You recall what temperature the slice recordings were done as compared to Vivo or whatever they are? Is in brief, but do you remember what temperature the source was?
So the recording, at least done in Vivo, it's done in the temperatures were matching between the two. This is this is done, but I can't remember the exact temperature. So these are fish that are in a tank, and the temperature of the water is maintained at the same level as when they actually perform the in vitro recording. So this is a collaboration that was done between us and Touboul lab.
Okay, so you're matching.
It's the same temperature. I got a few of this, yes. I can't remember off the top of my head what the temperature value, but that's what matching between. Okay, so that brings us to the first question that I'd like to answer. So what are the parameters that are contributing most, at least to the biophysical model, rather than the phenological model in generating heterogeneity? And are they matching with those that are generated by the simplified model? Okay, to do this, we divided parameters of the biophysical model into three classes: extrinsic, representing synaptic bombardment; intrinsic, which represents the conductances of the ion channels, or maybe their kinetics; and morphology, which is the coupling between the dendrite and the somat. And we asked the question, which one of those is actually contributing most to generating variability within the biophysical model? And this is essentially the results. What we did is that we developed this metric that quantifies the pairwise distance between the parameters that are associated to each model that is fitted to a cell. So I'm fitting each model to one cell, and then I'm quantifying the pairwise distance of these parameters that correspond to each one of those cells. And realize that these are 6 parameters that are contributing most to the variability within this population. Here I have about 300 cells that have been quantified at the same time. So you can see here, this is basically the synaptic inputs, how often you stimulate the cell with the glutamate, and this is the glutamate release, then the conductance of the escape channel, RPC concentration, and the NMDA conductance. So this is like multiplicative dynamics. And this is what parameter which corresponds to the morphology, the coupling strength between the soma and the dendrite. Okay. So essentially, this and this are consistent to what we made prediction in the simplified model, because this corresponds to synaptic bombardment, and this one corresponds to this flow adaptation that I have talked about earlier. So there is the result was consistent between the two. All right, now to validate this, to see the impact of this variability on the dynamics, we thought, let's actually visualize this in a low-dimensional space, because the parameter space is huge, and you cannot visualize it in such a large parameter space. So we need to do dimensionality reduction. So we use what we call principal component analysis. So essentially, we use the ISI distributions, features of the ISI distributions, to develop this to identify the principal components, and then find that you only need 3 principal components to explain more than 80% of the variance. Now, based on this, we mapped each cell into this 3 parameter space, 3 PC space. And this is essentially the distribution of all of the recordings within this 3 parameter 3 PC space. Okay. So this is a scatter plot that shows the distribution of the data. Then what we did is that we thought, I know that each one of those cells is fitted to a model. What I can do is that instead of course, it can capture the entire space if you fit each one of those cells using the entire parameter space. But now what I'll do, I'll make these 2 only vary while I keep everything else fixed. I don't change any of these parameters while I change these 2 together. Can it capture the volume that is occupied by all of these cells? Then I do the same thing, but I vary these green ones, intrinsic, and I keep everything else fixed. And then I do the last step, which is changing only this and keep everything else fixed. And this is the output. So as you can see, if I change only the extrinsic parameters, these two together, the volume occupied by the model fitting is significantly smaller than the ones occupied by the entire data set. In fact, it's even smaller if you change just the intrinsic inputs. So this one occupies more volume than this. And finally, if you change just kappa, you barely cover much of the volume. And if you do that comparison statistically, you'll see that the extrinsic properties kind of explain much more of the variance than what you see in the intrinsic properties and what you see in the morphological properties. So that emphasizes that the synaptic bombardment is the main driver in generating that variability within the data. Can we validate that experimentally? We are making predictions. Well, let's start with the first experimental setup. Remember the feedback I told you about? I have the pyramidal cells driving NP, and of course the NP also has a feedback into the pyramidal cells. So can we actually block that synaptic feedback into the pyramidal cells? And the answer is yes. You can actually block the sodium channel here using lidocaine. Lidocaine is applied on the NP, so you block the synaptic feedback coming back into the pyramidal cells in the ELL. What are the results in the PC space? In the PC space, this is control. Now with the block, the volume is much smaller. So you reduce the variability when you remove the feedback. To validate our prediction even more, we fitted each one of those cells. By the way, these cells are the same as these. They're identical. So we took these cells and we refit them using the model. And we asked the question, which one is generating actually most change as a result of going from one condition to the next? Which one of those parameters changed the most going from year to year? Well, as expected, the extrinsic parameters, the sigma and the lambda glue, which is the synaptic bombardment and the glutamate release, changed the most compared to any other parameter in the analysis to go from this condition to this condition. And of course, if you compare again the volume, once again, you'll see that in the block condition where you fit all the data, and if you just change the extrinsic properties, those ones are actually having less of a trouble to be closer to this than when you just change the intrinsic properties. So again, emphasizing that synaptic bombardment or synaptic inputs are key in generating that variability. Can we do the opposite? Can we enlarge the volume rather than decrease the volume with the block? And the answer is yes, you can do the same thing, but this time, what you do is that you apply serotonin. It's a neurotransmitter. What that neurotransmitter is known to do is to change the escape conductance. If you decrease the escape conductance, the neurons are going to fire at a higher rate because this is an output current. If you remove an inhibition, you're going to increase the firing rate of these neurons. So that's what we did. We added serotonin and looked at, again, in the PC space, what happens. This is in the control condition. This is in the serotonin condition. And as you can see now, the volume has become larger. You have a spread more in the PC space. Let's fit the model to here, fit the model to each one of those, and ask the question, which parameters are changing the most? And that's what you get. Remember I said the serotonin is known to change the SK channel? Well, SK conductance changed the most, as expected. So it's consistent with the expectation. But here's the surprising thing. It also affected synaptic inputs, the signal. That wasn't expected. But think about it. Pyramidal cells, you're adding serotonin, pyramidal cells can fire more. If they fire more, they're going to activate NP more. If they can activate NP more, the feedback is going to be higher. So the synaptic and the feedback and the synaptic feedback is going to be affected, and that's exactly what you see here. So it's actually not surprising to see that effect as well. So this is consistent with the data, and this was predicted, but it makes sense based on the connectivity that we have in the circuit. Okay, again, we quantified the volume just to emphasize, and you'll see that extrinsic is key, not just intrinsic. Oh, sorry, not just intrinsic, but extrinsic is key in generating that variability. Okay. All right. So now that we have answered that heterogeneity question and the differences between in vitro and in vivo, that brings me to my very last question. What role do bursts play? These neurons burst. But why are they doing this? Why do they switch between bursting and this isolated spiking events? And what role it plays, at least particularly in neural coding? So let me bring back what we know already about these weakly electric fish. As I said before, these fish have neurons on their skin. They're called electroreceptor afferents. They generate an electric field that allow them to communicate with conspecifics, with other fish, with predators, etc. And as you can see, each one of them have their own frequency. Each one of those fish will have their own frequency. Now, the interaction of two different frequencies generate these beats that you see there. Okay. Now, the interesting thing about these fish is that they sometimes alter their frequency, the frequency of the OAD produced by the electroreceptors. And that change in the frequency is called chirp. So you could basically, one fish could change their the frequency of their EOD, and that actually alters the beats that you see here. That could be actually located at any location along the beat. And you can see it here. Now that chirp could change in terms of amplitude, it could change in terms of width, it could also change in terms of the phase relative to the beat. So it could be at the top of the beat or at the bottom or anywhere in between. So there is a variability here that could be implemented within the chirp. No matter how much you vary that chirp, the fish still recognizes that this is a chirp and will respond exactly the same way. To give you an example, I just mentioned that to Jonathan earlier. I don't know if you've heard about the Jennifer Aniston neuron. You see a picture of Jennifer Aniston, different positions. You recognize that that person is always Jennifer Aniston. It's invariant. You know that that's the same person, even though it has different angles. And there are specific neurons that respond to those pictures of Jennifer Aniston. In fact, they're called the Jennifer Aniston neurons in the paper that was published on this topic. So the fish is doing exactly the same thing. They are reacting invariantly to these chirps, no matter what they are. In fact, if you look at the correlation, so here, these are different variations of the chirp in terms of phase, amplitude, or width. And if you look at the correlation between these chirps, there's no correlation at all. You don't see any correlation between the amplitude, the phase, and duration, chirp waveform. No matter what you do, they're always uncorrelated. But still, the fish is still responding the same way to that chirp and recognizing it as one of the same thing. So you can see that I'm heading in one direction to say that bursts are related to this. So let me show you why. Okay, so let's say that you have a chirp. This is a chirp here. And you look at the response of a neuron 1 and a neuron 2. And this is essentially, a chirp superimposed on a beat. And you see the firing of these neurons there. And this is another chirp as well. Now you can isolate the burst from the isolated spikes, separate between these two. Now these are the bursts correspond to neuron 1, and these are the isolated spikes corresponding to neuron 1, and you do the same thing, or neuron 2, sorry. And then you do the same thing for the isolated spikes. You could separate between these two. And of course, you could do that for any neuron in the recording. Then you ask the question, what happens if I sum up all the responses of all of these neurons to different chirps? One that is changing the phase, one that is changing the width, one that is changing the amplitude. Now, this is summing up all the responses of the neurons. Now, what I do here, I isolate the burst, and then I isolate the single spikes. You'll see that regardless of the type of chirp that I have, the burst always generates the same response. It kind of peaks right when the chirp happens. Unlike the isolated spikes, they're not doing anything. There's quite a bit of variability between the two conditions. The bursts are the only ones that are reacting invariantly to these chirps. Now, if you look at the ISI distributions, in fact, for all spikes versus bursts versus isolated spikes, and you look at the coefficient of variation, correlation coefficient using Pearson correlation, you'll see that it's highly correlated when you talk about the burst, unlike what you have with the isolated spikes or all spikes. So it seems that bursts are playing a fundamental role in generating that invariant coding in these electric fish. And that happens at the population level, because if you do it at the single neuron level, over all trials, you don't see anything. But if you do it at 73 neurons within a network, you see that highly correlated here, unlike the isolated spikes, and the overall you see still a response because of the contribution of the burst. And if you actually look at it continuously between one neuron all the way to 73 neurons, you see that the invariant coding is maximized as you account for the bursting. Now we have a mathematical model to do this. I can actually use it to see what impact bursting has, because I can manipulate bursting. Remember, I have a two-compartment model, and that compartment model can generate ghost bursting. Can I basically alter the bursting? Can I make it less frequent or more frequent? The answer is yes. I can target the soluble channel. I can't remember which one is soluble channel, but one of those. I can basically target the conductance of the soluble channel, the dendrites, and I can actually alter the amount of bursting that happens in these neurons. So this is what we call GNAD, the conductance of the sodium in the dendrites. And what you see here is that as you change the burst fraction within a recording, the number of bursts within a recording, and you look at different GNAD conductances, you see which one of those would maximize the correlation or the invariant coding. You notice that at intermediate level of GNAD, you maximize it here. If GNAD is too low, you don't have much bursting, you don't have much, it doesn't actually maximize it. If GNAD is too high, it also doesn't maximize it. It actually brings it down as well. So there's a Goldilocks zone of GNAD values that will maximize invariant coding, because here you have optimal number of bursting burst firing that will allow you to have this feature. Remember, this is a prediction of the model. We did not fit this to the data, this feature to the data. But guess what? If we compare that value to the data, it's a perfect match. The data is here for the GNAD, and this is where the maximum occurs. We did not use that feature for fitting at all. It was purely predicted, and there's an identical match between the two. Okay? All right. Yeah, go ahead.
So the burst happened at this church. What happened to the rate of isolated spikes? Does it increase in the same window?
Yes, yeah, it will increase. So if GNAD is too low, most of the firing would be isolated spikes. If it's too high, it would be mostly bursting.
So I guess that's the opposite of what I'm asking, because I'm asking in that chirp window, you see that the increase in the burst, but the rate of isolated spikes, does it also increase?
No, it doesn't. It's they're negatively correlated.
I see.
So one will increase, the other one will decrease, and vice versa. Yeah. I was hoping to ask another question, but you didn't. What is the role of isolated spikes?
Yeah.
What role? I mean, here in invariant coding, what does the isolated spikes do? It's the opposite. I have that as supplementary figure. I'll show you that towards the end if you want, if you're interested. I don't have it in my presentation here. Anyhow, this is our conclusion. So we managed through this study to basically compare in vitro to in vivo dynamics and see how they are translate to each other, what the role, for example, of the escape channel in this phenomenon. And then we basically looked at heterogeneity as well, how that accounted for, what actually causes that heterogeneity in neuron responses. We validated some of these predictions experimentally. And finally, we highlighted the role of nurse firing and maximizing invariant coding. For references, here are the references. There are 3 papers. This one is going to appear over the next couple of days. I accepted about 2 weeks ago. So it should appear soon, but these have been already published. And finally, I'd like to highlight my team, particularly Amin, who's now a postdoc at the University of Calgary. He's right there. It's an old picture, but that's him as well. He's the one who did most of this work, and of course the funding that supplied the money to support this project. I'm happy to take any questions you have. Now, coming to your question here, I just want to show you it increases discriminability. So invariance and discriminability. So isolated spikes allow you to discriminate. It's Jennifer Aniston, but Jennifer Aniston is standing this way versus standing this way. So you could tell that this is the same person, but at different locations. And this is what actually we did. So you could see that discriminability is higher compared to bursting for isolated spikes. It doesn't do it perfectly. It's not matching the one that you have for the entire recording, but it's higher than bursting.
So what do you look at in that case in the isolated spike? Like, what do you quantify for the isolated spikes in this case? Like, what do you discriminate with in the isolated spike?
Do you discriminate? Oh, I mean, at least in the context of chirps, you discriminate that the amplitude is larger.
For the chirps.
For the chirps.
But isolated spikes, you're looking at just like rate, or what are you looking at in the isolated spikes to say, well, that changes in a way that is relevant to the change in.
Oh, I see. Like, what is the signature?
Yes.
We did not look at this. Sorry. I understand. That's a very good question. Like, what is the signature within the isolated spikes that distinguish amplitude versus phase versus? That's an excellent question. We did not look into this. Yeah. Now that would be a much harder question. Yeah.
So I'm putting this together for the whole history of the general avoidance response. And anything that's been done with that, and having trouble not mapping the 2 sets of literatures.
Okay. Between which one and which one? Sorry,
So
I
the
didn't hear.
general avoidance response. The whole literature about the general avoidance response.
Not familiar with this. Sorry.
Okay, these guys shift their frequencies according to what's happening to the other fish.
Okay.
So, there's 12, we have to figure this out.
Yeah.
There's this beautiful story in these guys that they produce given frequency, and then it's it, and then if you give them that another animal plus a similar frequency, both animals change their frequencies.
Okay.
This is the last one. Go back to that slide where we have the
The oscillation, yeah.
So there, the peak frequencies, each animal has a characteristic frequency,
Yes.
and they use that and when you
Okay.
things. Yeah, but what they do is if another fish comes with a very similar frequency, that would confuse everything.
Yes.
So the one that's higher goes higher, and the one that's lower goes lower. So they created a larger difference between the two. And so.
I mean, I know that people have looked into this quite a bit, analyzing the dynamics of electroreceptor afferents that generally these EODs. So I'm not too familiar with the literature on this thing, but we are basically trying to, we're trying to understand the dynamics at a bit of a higher level at the ELL as opposed to in the electric receptor affluent level. Does that make sense?
Yeah, well, but this is doing it so it can behave accordingly, so
Yeah.
it's not, it's changing its whole brain activity, right? That's fine.
I understand what you mean, but I think actually, the point that I want to say is that if you're worried about the fact that, you know, these fish would change their frequency of their EOD, I would argue that the pipeline that can be applied to, that developed here, could be applied to this, the set of neurons that are generating these frequencies. And I don't believe that the conclusion would be any different. So we're basically arguing.
But in other words, you have a fish and you have a population that frequencies is pretty similar.
Yeah.
We'll just have to change just a little bit.
I get what you mean. So even if they alter their frequency, and you're arguing that that could basically affect the neural dynamics upstream, like in BLL or even high brain regions, we can repeat the same sort of analysis that we have here on these neurons with this neural a new data set. And I would argue that we could reach the same conclusion in terms of role of the SK channel, role of synaptic bombardment in generating these differences between in vivo and in vitro and its heterogeneity that we present here. So I don't think the conclusion would change much as a result of that. It changed in the frequency of EOD.
So bursts happen at these chirps, but how specific are bursts to these chirps compared to other environmental or behavioral changes? For example, is it only happening when there's a chirp, or if the fish changes its behavior from not swimming to swimming very fast? So yeah, does this make sense to you as a question? Is it something totally specific to chirps and the detection, or is it just the representative of a change in the environment, or in the behavior, or in the?
Actually, I don't have that result, unfortunately. I mean, you could see here that at least when it comes to the correlation, you're not actually, reaching one. You're not definitely at one. So there are other factors that could be also contributing to the variance. But here, the bursting is contributing significantly. I mean, the population size is 73. That's the maximum number of neurons that we consider. But you could see that it's also kind of plateauing. So there could be other factors that could be contributing to invariance as well. So that could be related to factors that are related to environment that could make the animal adapt.
Yeah, what I'm trying to get at is this machinery, how specialized it is, because a chirp, at the end of the day, the way I'm seeing it is a change in the environment of that fish, right? So
it's communication between two fish.
So it's a change in the communication.
Yes,
right?
Yeah.
So in other things, like the simple thing I can think of is how fast the fish is swimming. Which would be an internally generated motor behavior in that case. If at the points in which there is a sudden shift in the fish's behavior, do we know if we also see a burst? Is it a generalized mechanism for detecting changes in multiple planes?
Oh, I see what you mean. Well, yeah, that could be a very good question. I mean, we did not explore the role of bursting in adaptation, for example, adapting to different environments. That could be a valid question to explore, but we did not do that here. You're right.